want to thank the organizers and the chairpersons, and then I'll move on to it. So the way I'm going to talk about is just a little bit what reactive oxygen species are, give information from humans, then we go to the cells, then we go to the rat that I work with, that you should be familiar by now that I have given 2 talks, and then in the end I'll try to see how all this information has led me to believe that reactive oxygen species may be involved in stone formation. So the part is the reactive oxygen species, and I'm sure that everybody has heard so much about antioxidants that we know, but the part is that we don't realize this thing. Everybody thinks about reactive oxygen species and oxidative stress, but they don't realize that reactive oxygen species are very significant for actions throughout the body. You look at any physiology book, go ahead and look at it, they are always involved. But the part is that whenever they are formed at exactly the place they are needed. They don't get away from it. But when there are too many of them, that they move out from where they are needed, then they become a problem. So the reactive oxygen species formation is physiological, but the overproduction. But on the other hand, as I said, these are physiological things, but so the body is prepared to have a little more of them. They have their own system of antioxidants. So the body maintains that homeostasis between production of reactive oxygen species, and if some of them want to get away from it, they want to be used up. But once either the body's own antioxidative medicine comes down, or there are more are produced and they get away from where they are, then there is a development of oxidative stress. And oxidative stress, the way you look up, you try to find out reactive oxygen species, which are very difficult to study. So what happens is you look at the products of the lipid peroxidation generally, you're looking, those are called things like melandialdehyde, isoprostane, and et cetera, and et cetera. So as I said, while generation of large amount of ROS may play a major role in tissue injury or death, but regulated generation of low concentration of reactive oxygen species may represent a second messenger system for generation of macromolecules involved in nephrolithiasis. So that is because we are very familiar with it, that there are inhibitors there. So how are those inhibitors formed? As I gave a lecture yesterday on the subject, the reactive oxygen species play a significant role in the production of macromolecules like osteopontin, macromolecules like temharsval protein, bicunine, prothrombin, all those things which we think as inhibitors. So those are a very significant thing. So now, as far as the human studies is concerned, there it is. This came out from the Thailand. It's published in 2005 study here. And in this case, if you look at this is the melandialdehyde. Melandialdehyde on this side, they're showing the difference between the stone formers and non-stone formers. When you start looking in the urine, you will see the products of lipid peroxidation in kidney stone formers, and there are a number of other studies. I'm just giving you one example that I have here. Then in this particular case, from the same paper, you can look at these. There is a difference between the stone formers and the non-stone formers in the amount of these different activities. And also, you look at NAG, which is almost all the nephrologists know these things, that they're markers of nephrological injury. So you can see that there is an injury there is an increase in the lipid byproducts in the stone formers. Now we go there, that was just looking at it. Then the other source of information about the involvement of reactive oxygen species is this study that came out in Journal of Endourology in 2011. They were looking at the antioxidants and the self-reported history of kidney stones, and they did with ENHANCE, so you know there are thousands and thousands of patients in this group, and after adjusting for covariants, mean levels of alpha carotene, beta carotene, and beta cryptoxanthine were significantly lower in those with kidney stones. When analyzed by quartile, higher serum levels of beta carotene and beta cryptoxanthine trended towards a decreasing prevalence of stones, indicating that the highest level of these antioxidants may protect against the formation of kidney stones. Lower levels of alpha carotene, beta carotene, and beta cryptoxanthine are associated with a history of kidney stones and may indicate a role of these antioxidants in preventing stone formation. Another study was statin use. That statin use is pretty common. In this particular study, they looked at hyperlipidemia, statin use, and stone recurrence. That came out again in Journal of Urology. These were Armed Forces Health longitudinal study. So there's a big database in, and what they found was that statin use was associated with a significant decreased risk of urinary stone formation compared with non-user, even after adjusting for age, gender, obesity, diabetes, hypertension, and cardiovascular diseases. Then the best, at least from my point of view, is the DASH diet. DASH diet is one of those diets. There's a lot of people who have hypertensive and other diseases, the doctors will put them on it. So there are 3 different studies. This Taylor et al.'s 2009 prospective study, health professionals follow-up. You look at the numbers, 45,000, and then another study, 94,000. DASH-style diet was associated with reduced risk of kidney stone formation. Then, so you will say, what is DASH diet? What does it have got to do with anything with the reactive oxygen species or antioxidant? Then, low sodium DASH diet reduces oxidative stress and improves vascular function in salt-sensitive humans. Then another study from hypertension, DASH combination diet raises antioxidant capacity, lowers blood pressure, and reduces oxidative stress in free-living hypertensive patients. I don't know what free living was, something like free range. Anyway, so then you start looking at all these diseases that DASH diet and other things happen. So the urolithiasis and chronic kidney disease. Chronic kidney disease is the loss of kidney function. Diabetes and hypertension are the most common causes and account for most CKD. Urolithiasis is also known to be associated with CKD directly or perhaps through diabetes and hypertension. And here, when you start looking at all these diseases, whether it is nephrolithiasis, chronic kidney disease, diabetes mellitus, obesity, metabolic syndrome, hypertension, if you look at the pathogenesis, particularly in the experimental models, what you find that all of them have the production of reactive oxygen species is common to them. The other common thing is that they have oxidative stress, but then the third component of all these things is inflammation. So because these diseases, they all have oxidative stress, they all inflammation. So it was, in my mind, the part is that if one disease causes a oxidative stress, it can affect the other thing. For example, a hypertensive patient, a normally slightly raised oxalate may not induce kidney stone, but if a hypertensive patient has a slightly increased oxalate or lower citrate or other conditions which a normal person will live with, but if they have other comorbidities, that will lead to nephrolithiasis. Then here, this is from our tissue culture work. When we find it that when the cells were exposed to the calcium oxalate crystals and oxalate exposure, they all produce reactive oxygen species. And when you treat them with antioxidants like catalase, on this side you see this is calcium oxalate monohydrate, here it is calcium oxalate monohydrate with catalase. So when you treat these cells, you expose the cells to oxalate or calcium oxalate crystals, there is a production of reactive oxygen species. But on the other hand, when you treat them with catalase, and in this particular case, we are looking not directly for the reactive oxygen species, we are looking at monocyte chemoattractant protein 1. So here we are not saying the oxidative stress, we are actually saying inflammation, because monocyte chemoattractant protein 1 is, as the name implies, it is a part of the inflammatory cascade. The other thing is that in this particular case, we were looking at the production of superoxide, which is a reactive oxygen species, which leads to oxidative stress, and then we tried to lower the production of superoxide through the inhibiting NADPH oxidase, which we think is responsible for the production. And in this particular case, we started looking at the osteopontin production. And in here, you can actually see this thing. There is a production. There is this particular compound, which I say DPI. DPI is an inhibitor of NADPH oxidase. So when you use the inhibitor for NADPH oxidase, we reduce the production of superoxide, which is shown here, and then it leads to lessening and it affects the production of osteopontin. This is another study. In this particular case, again, we are talking about the NADPH oxidase. In this particular case, we had MDCK cells, exposed them to oxalate and calcium oxalate crystals, and then started looking at the activation of NADPH oxidase, and these crystals increases that. In this case, which is from a different lab, they were just looking at the production of superoxide in the presence of calcium oxalate crystals and oxalate, and it turns out to be that there is a, whenever you get the cells, you expose them to the oxalate or calcium oxalate crystals, there is increased production of superoxides. So now, those were tissue culture thing. Then you move on to the animal models, which is generally the rat. And what, as I have said before, what happens in the rat model, you induce hyperoxaluria, they produce calcium oxalate crystal deposition. But if you also treat them with antioxidants, like I showed you before, apocynin, and some other like vitamin E, then what happens, crystal deposition is reduced. In this particular case, the thing was treated with apocynin, which is an antioxidant and NADPH oxidase inhibitor, and you can see the difference. This is untreated by apocynin, here is treated one, and it reduces the deposition. We'll move on. In this particular case, this is from Dr. Selvin and Dr. Manon, and here they used vitamin E. And in this particular case was very interesting that they were completely, by treating their rats with vitamin E, they were able to completely stop crystal deposition in a hyperoxaluric rat. And this is actually the, let's see, this particular case is the next one. Here again they use, this is a rat model of hyperoxaluria, and in this particular case they gave them atorvastatin. So it is another statin which is an antioxidant, and what happens is here, this is without the treatment with the statin, this is with the treatment, the crystal deposition goes down. We'll keep moving. So now coming back to the, how does it come up with the Rundle's plaque and plugs and the stone formation. So if you look at these two things, we have been listening to these talks for a while, so I don't have to go into too much detail. Here is the stone which is attached to the Rundle's plaque, and here it is forming into the collecting duct, terminal collecting duct, as a plug. So plaque and plug, there are two different ways for kidney stone formation of calcium oxalate. So it's my theory is this, that if you go through this thing, that if there is a problem with the calcium phosphate, citrate, oxalate, crystals in tubular fluid, renal papillary tubular epithelium goes under stress, reactive oxygen species are produced, there is a dysregulated signaling which activates a number of transcription factors that converts the epithelial differentiation into a bone type producing cells, and then they start making calcium phosphate deposition on the basal side. That calcium phosphate deposit, if it is happening at the collecting duct or in the loop of Henle, will turn into a Rundle's plaque, and so this way the reactive oxygen species become involved with that Rundle's plaque thing. And then you look on the other side, the plug can very easily be formed. There is hyperoxaluria, hypocitraturia, crystallization is oxidative stress, pathways, there is a crystal cell death, membranes, nucleation, and it is happening in the lumen, and it turns into a formation of a plug at the plaque. And here, I mean, if you look at this, these are some of them. They look like they were formed at the plug. So basically, that's how I think they may be involved. Thank you very much.